Good afternoon, all. Today, my topic is based on human protein replacement that include insulin, human growth hormone, and hemophilia. Any defect in a gene produce incorrect protein or no protein at all. This defective protein may cause disease. Thus, gene defect results in genetically linked disease. The recombinant DNA technology employed to produce human protein that can be used for the treatment of genetically linked disease, and this procedure is mainly known as human protein replacement. Next, about insulin. Insulin is a polypeptide hormone produced by the beta cells of pancreatic islets of Langerhans. These insulin mainly regulate blood glucose level and important in the metabolism of protein, lipid, and they display mitogenic activity. The main target of the insulin include skeletal muscle fibers, hepatocytes, and adipocytes. Insulin responds to the absorption of glucose and other nutrients in a number of ways. And first point is it stimulates glucose transport that also important in transport of amino acid, potassium ions, and other nutrients into the cell. Therefore, they thus reduce their blood concentration. Second one, it stimulates intracellular biosynthetic or anabolic pathway such as glycogen synthesis, which help to convert the nutrients into storage form in the cells. Third one, it inhibits catabolic pathways such as glycogenolysis. Glycogenolysis means breakdown of glycogen to glucose. Therefore, this insulin inhibit the catabolic pathway such as glycogenolysis. Fourth one, to stimulate protein and DNA synthesis. Next one, diabetes mellitus. This diabetes mellitus mainly occurs due to the failure of the body to synthesize sufficient insulin, results in the development of insulin-dependent diabetes mellitus or IDDM. They are also known as type 1 diabetes or juvenile onset diabetes. In diabetic patient, the ATP production is mostly in depressed condition because there occurs a mitochondrial dysfunction. As mitochondria is energy currency of the cell, therefore, the dysfunction in the mitochondria results in ATP production. Therefore, in diabetic patient, the ATP production will be depressed condition. This IDDM is caused by T cell mediated autoimmune destruction of the insulin that producing beta pancreatic islet cells. Next one, insulin molecule. It is first identified as anti-diabetic factor. This mature insulin is a dimeric structure synthesized as a single polypeptide precursor that is a pre-pro-insulin. The insulin molecule mainly consists of 108 amino acid polypeptide that contain 23 amino acid signal sequence at its amino terminal end. The proinsulin is proteolytically processed, that is secretory granules which is mostly coated with clarithin, and they yield mature insulin and 34 amino acid connecting peptide known as C-peptide. The C-peptide is further proteolytically modified by the removal of dipeptide from each of its ends. Mature insulin, two polypeptide chains connected by two interchain disulfide linkage. We can see in the diagram here. That is, this is the A chain and this is the B chain and this is the C peptide. Therefore, the C peptide undergo proteolysis and form a mature insulin, and this is also known as A chain and B chain. And this A chain consists of 21 amino acid residue and this B chain consists of 30 residue. Therefore, these two chains are linked together by interchain disulfide linkage. Next one is insulin receptor and signal transduction. This insulin receptor is a tetrameric integral glycoprotein that consists of two 735 amino acid alpha chain and two 620 amino acid beta chain. The both chain is joined together by disulfide linkages. Here in alpha chain, this entirely on the extracellular side of the plasma membrane and they contain cysteine-rich insulin domain. Beta chain composed of three regions, that is extracellular domain, transmembrane domain, and large cytoplasmic domain, which displays tyrosine kinase activity. Proteolytic digestion of alpha subunit results in activation of kinase activity. This is a diagram of this is alpha subunit. This is a beta subunit, and they possess in beta subunit they possess three regions, that is juxtamembrane domain, tyrosine kinase domain, and C-terminal domain. Here, three positions present in the cytoplasmic domain of the insulin in beta subunit. Binding of the insulin promote autophosphorylation of the beta subunit, and these phosphate group are attached to three tyrosine residues. That is, the insulin is binded here, and these undergo autophosphorylation and form phosphate groups are get added to the tyrosine residue. Activation of beta subunit tyrose kinase activity results in the phosphorylation of intracellular substrate, which trigger inducing mitogenic and metabolic effects. That is, due to this activation of the for autophosphorylation, two pathways get activated. That is MAP kinase activity and PI3 kinase activity. This MAP kinase activity results in mitogenic effect, and this PI3 kinase activity results in metabolic effects. Next, the production of human insulin by rDNA technology. Insulin was first approved in 1982. The initial approach was inserting nucleotide sequence coding for the insulin A and B gene into two different E. coli cells. And these A and B genes are incubated together under oxidizing condition, and they promote interchain disulfide bond formation, forming human insulin CRB. This is the first procedure. An alternative method is produced by Eli Lilly. That is, she produced in inserting nucleotide sequence coding for the insulin A and B gene into recombinant E. coli. Gene for insulin A and B chain are inserted separately to the plasmids of two different E. coli structure. In former procedure, they are oxidized together or incubated together in oxidizing condition, but in this procedure, they are inserted separately to two different E. coli structure. Lac operon system that consists of inducer, promoter, operator gene and structural gene set for beta galactosidase is used for expression. The presence of lactose in the culture medium induces the synthesis of insulin A and B gene in separate culture. As I said that presence of the lactose in this medium promotes synthesis of insulin A and B gene in separate culture. And then the formed insulin can be isolated, purified and joined together to form a human insulin PRB. As the human insulin PRB is a recombinant product produced, it is very identical in sequence, and any impurities present will be host derived and potentially highly immunogenic. As it possesses impurities and is highly immunogenic, purification of the product must be undertaken. This is a schematic diagram of the human insulin production of PRB. This is a lac operon and G for insulin A and B chain, and this transform into E. coli, and this separated A and B chain formed, and this then joined together by interchain disulfide linkage and form mature human insulin. Next, purification process. A cleanup process scale RP-HPLC has been introduced into production of human insulin PRB. RP-HPLC means reverse phase high performance liquid chromatography, and this process internal volume of 80 liter or more up to 1200 gram insulin may be loaded during a single purification run. Separation is achieved using an acidic mobile phase that is set at below insulin PI value. PI value means isoelectric point of insulin below 5.3 in order to keep it fully in a solution. This insulin is usually loaded in the water-rich acidic mobile phase, followed by gradient elution using acetonitrile, that is 15 to 30%. The starting material loaded onto the column is fairly pure, that is 92%, and this step yields a final product of approximately 99% of purity. Over 95% of insulin activity loaded onto the column can be recovered. Single column takes about 1 hour. The RP-HPLC polishing step not only removes E. coli derivative but also separates modified insulin derivative from the native insulin product. Therefore, the resultant low level of impurity in this insulin preparation failed to elicit any significant response in diabetic recipients. That's we show that modified insulin derivatives also get removed during purification. As a result, the insulin response will fail in diabetic patient to significant or to active the response of an insulin due to the removal of the modified insulin derivatives. Is a schematic diagram. Undergo fermentation and form crude proinsulin, and they undergo purification, crystallization and form partially purified insulin. And Then they form crude insulin or mature insulin. They undergo ion change, enzyme filtration, and form purified insulin. And they undergo RPHPLC and form highly purified insulin. This is the sorry. This is the HPLC column. Next one, the formulation of insulin product. Fast acting insulin, that is short, elevated blood insulin concentration quickly after administration. That is fast acting insulin when administered into the skin, they undergo elevated blood insulin concentration. Slow acting insulin, that is long acting, long time duration, that enters circulation slowly from the ejection site. Insulin secretion peaks approximately 1 hour after meal and base level again within 2 hours. In order to prolong the duration of insulin action, soluble insulin may be formulated to generate insulin suspension. This can be achieved in two ways. First is addition of zinc in order to promote zinc insulin crystal growth. Second one, addition of a protein to which insulin will complex and form, in which insulin will only be slowly released. The protein normally used are protamines, which are basic polypeptide naturally found in association with nucleic acid in the sperm of several species of fish. These two procedures are done to increase the duration of the insulin action. Depending upon the relative molar ratio of insulin protamine used, resulting long-acting insulin generated are termed as protamine zinc insulin or isophane insulin. Next one, engineered insulin. The major aim of generating such engineered insulin analog include identification of the insulin with altered pharmacokinetic properties such as fast-acting and slow-acting insulin, and identification of the superpotent insulin forms. Fast-acting insulin include human insulin Lispro or Humalog, which is the first fast-acting engineered insulin. Insulin aspart is the second fast-acting engineered insulin. They differ from native human insulin, that is, proline B28 residue has been replaced by aspartic acid. In native human insulin, they possess proline B28, and in this fast-acting insulin, it is replaced by aspartic acid. Longer-acting insulin exhibit plasma life up to 35 hours. Levomer differ from native human insulin, devoid of threonine B32 330 residue. Insulin glargine differ from the C-terminal asparagine residue of the A chain that has been replaced by glycine residue, and the B chain has elongated by 2 arginine residue. Alteration of native protein amino acid render the engineered product immunogenic. Next one, human growth hormone. Human growth hormone or somatotropin is a polypeptide hormone synthesized in anterior pituitary. They promote normal body growth, lactation, and various aspects of cellular metabolism. The mature human growth hormone consists of 191 amino acid residue with a molecular mass of 22 kDa and two characteristic intrachain disulfide linkages. This human growth hormone mRNA undergoes splicing and yields shortened growth hormone molecule about 20 kDa, which displays biological activities indistinguishable from 22 kDa species. That is, when this undergoes splicing, the molecular mass becomes shortened to 20 kDa from 22 kDa. Therefore, they displays biological activity that is indistinguishable from 22 kDa species. Human growth hormone synthesis and release from the pituitary is regulated by two peptide hypothalamic factors. First one is growth hormone releasing hormone, that is GHRH, also known as GH releasing factor GHRF or somatorelin. Second one, growth hormone releasing inhibiting hormone, growth GHRIH or somatostatin. Major influence on body growth is mediated indirectly by IGF-1, GHRH, GHRIH, thus form a hormonal axis. Growth hormone receptor. Growth hormone induces its biological effect, that is, they bind to specific cell receptor, human receptor, a single chain 620 amino acid transmembrane polypeptide. In humans, soluble growth hormone binding protein GHPP are generated by enzymatic cleavage of the integral membrane receptor, releasing growth hormone binding extracellular domain. Here, they possess growth hormone and growth hormone inhibiting hormone. They both reach into pituitary gland, and from there they release growth hormone, and major of them release in liver, and they release IGF factor 1 that mainly affect the growth promoting effect, and other one indirect effect on many cells. Biological effect of growth hormone. They displace an anabolic activity. They stimulate the growth of bone, muscles, cartilage cells directly. The binding of growth hormone to its hepatic receptor results in the synthesis of release of IGF-1, which mediate the most growth hormone promoting activity. That I said earlier, the deficiency in the secretion of human growth hormone results in pituitary dwarfism. Overproduction of human growth hormone results in gigantism, acromegaly. There is a condition characterized by enlarged hands and feet. Therapeutic use of growth hormone. Human growth hormone extracted from human pituitary glands are used to treat pituitary dwarfism. They are effective in treatment of short stature, including Turner syndrome, idiopathic short stature, and chronic renal failure. The recombinant growth hormone was produced in E. coli. This recombinant preparation differ from the native human hormone only that they contain extra methionine residue. That is, they differ from the native human hormone from that they contain extra methionine residue in the beginning of the gene. Several growth hormones include gonadotropins, that is involved in the development of sexual characteristics, follicle stimulating hormone and luteinizing hormone. They involve development of male and female reproductive function. Androgen, that is sex hormone in male, possesses Leydig cell for sperm synthesis. Estrogen, involved in the growth and maturation of female reproductive system, female behavior, effect, etc. Next one, factor 8 hemophilia. Hemophilia A is an X-linked recessive disorder caused by the deficiency of factor 8. Von Willebrand disease disorder that is caused by the defect in the factor 8 complex. Factor 8, usually purified from the blood, consists of two gene products, that is factor 8 and multiple copies of von Willebrand factor. That is factor 8 consists of two gene product, factor 8 and multiple copies of von Willebrand factor. Factor 8 yield two fragment, that is amino terminal 90 kDa polypeptide and 80 kDa carboxyl terminal polypeptide. These associate non-covalently to produce the mature factor, factor 8C. The mature factor 8C is then released into plasma where it associates with the multiple copies of VWF forming factor, factor 8C is to VWF. That is, in a healthy individuals, we can see that in liver cell they release factor 8C and endothelial cell they release VWF factor. They form a complex, factor 8 complex, that is 8C is to VWF. These process in a healthy individuals. The person who are affected in hemophilia, from the liver cells, this factor 8C will not be released, and endothelial cell they release VWF factor. Therefore, the deficiency of the factor 8C results in the hemophilia. And in third condition, the person who is affected in their own Willebrand disease, the liver cell release factor 8C, but endothelial cell does not release VWF factor. Due to the lower level of VWF factor or absence of VWF factor, the factor 8C undergo rapid degradation of 8C in the blood. Therefore, the deficiency of the VWF factor results in the von Willebrand disease. This is three condition in a person occurs. Production of factor 8. Factor 8 is purified from blood donation to screen the presence of hepatitis A and HIV. Various precipitation process has been done. First, they undergo filter sterilization, then form finished product, then they are packed into containers, then freeze-dried and sealed under vacuum. This is a purification or precipitation process. Although these precipitations are relatively crude, lower levels of other plasma membrane and modern preparations are done, and use of immunochromatography has been widespread. Several companies had expressed cDNA coding for human factor 8:C. Recombinant factor generally contains only 8:3 devoid of VWF. Both preclinical and clinical studies have shown that administration of this product to patients suffering from hemophilia A is equally effective as administering blood-derived factor 8 complex. Researchers are also attempting to develop modified form of A2C by exhibiting extended half-life. Thank you.